Hi, welcome to the channel again. This video is a Biology Cape Unit 1 video where I'll be just discussing and sharing some notes on Objective 1, 2, and 3 on the syllabus, which is water, its properties and structure, and looking at polymers and looking at the first biomolecule, which is a carbohydrate, right? I don't know where students in the secular school have reached, right? But this video is just to assist you. You could look at this video and augment your notes of what was taught to you, right? So if you're looking at the screen, you would be seeing objective one is the structure and properties of water. And we want to go through that today. I want to also talk about polymers and also your first biomolecule, carbohydrates, and look at the types of carbohydrates and look at your first type of carbohydrates, the monosaccharides, and look at glucose. All right. It's the same approach that I will do for water and for glucose is the same that I will do in the preceding videos for sucrose, starch, glycogen, triglycerides, phospholipids, your protein, your amino acids, and also the tests for reducing and non-reducing sugars, lipids, protein, and starch, right? So it is just for the preceding videos and these videos is just to assist you to make sure that objective by objective in the syllabus you're covering it and you're understanding it, right? So it doesn't matter where you reach, you could just look at the video and augment your notes from what you already taught in school, right? So that is the objective 1 to 4 there in the syllabus, where you see in water, polymers, glucose, and then the structure of sucrose. And then we have your starch and glycogen. Then we have your triglyceride, your phospholipids, the amino acids, tests for certain biomolecules. And then you have your protein and the structure of certain proteins within organisms, right? And their function. So that is with the aspects of biochemistry. Following this in module 1 is the cell structure and function, membrane structure and function, and then lastly enzymes before you go into module 2, right, which is your genetics and your variation and natural selection. And then your module 3, reproductive biology, right? So for CAPE students who do in unit 1, the new stuff is basically um, your aspects of your biochemistry, right? The what will be familiar from CSEC is module 2 with your genetics, variation, and natural selection, right? There'll just be a little additional component there with your Punnett square and so on, and looking at certain null hypothesis and so on, right? Just additionally, but yeah, once you have a good foundation with your CSEC in genetics and variation and natural selection, you'll hold your N in module 2. And your reproductive biology is just looking a little more closer at the meat ending of the human and so on, right? So your module 2 and 3, you'll be very familiar with the concepts inside there and then just looking a little more in depth in it. So the only new thing in module 1 is these aspects of your biochemistry and your biomolecules, right? And to understand these molecules, you heard about it in CSEC, is now to understand the structure and the function in the body, right? So the first thing we're going into is your water, right? So if you look at this here, we see in that water, um, we all know we need water, right, to survive, right? Um, and we see in basically what you have to take out for your biology concept here is the structure of water. And we already know from CSEC chemistry, the bonding in water is covalent bonding between the hydrogen and the oxygen. right? And we know covalent is sharing of electrons. And we was also taught in CSEC chemistry in terms of why your water molecule is polar, because you will have that oxygen being more electronegative and pulling electrons towards it, away from the hydrogens. So then you have part of the water molecule being partially negative and then part being partially positive. So it makes it polar, right? And that is a major factor for water being a universal solvent, right? And also it meets the criteria from CSEC chemistry also for hydrogen bonding. Because once you have a electronegative atom with lone pairs, you could have an hydrogen in it, you could have hydrogen bonding happening, right? So this is just talking about this first slide here, just talking about um, the bonding involving water and how it is polar, right? And I explained that just now. If we look at this one here, this is the covalent bond. Right? So you have your covalent bonding between your hydrogen and your oxygen, hydrogen and oxygen. You have two lone pairs of electrons up here, right? And if you're watching the shape here, you'll want to know why this thing is not straight, right? It's because these lone pairs of electrons up here would be pushing or repelling the bonded electrons. So that is why it is pushing down these hydrogens that are bonded to the oxygen to this angle, right? It reached this angle here now because now you'll have repulsion happening with these two partially positive here, right? So you have this bend shape. So the water molecule is the shape is known as a bent molecule with that angle there of 105 degrees, right? So this diagram is just to show you how the hydrogens are bonded and how the electronegative oxygen is pulling electrons from the hydrogen, so it makes it partially positive, right? And you get that polar nature. Right, so hydrogen bonds, remember from CSEC chemistry, would know that for hydrogen bonding to take place, the molecule or the compound must have an electronegative atom, lone pairs of electrons and hydrogen, right? So water could go through that. It is a good candidate for that. So hydrogen bonds form between the water molecules. As a result, the polarity of water hydrogen bonds form between positive and negatively charged regions of adjacent water molecules. So the whole structure of water, so if we have a beaker of water, why we see that consistency is because of this hydrogen bonding between molecules of the water, right? And if you look at the second point there, hydrogen bonds are weak. So when they are few, so they are consistently breaking and reforming. However, when they are in large numbers, present a form of a strong structure, right? And we will talk about that latter line there when we look at the cohesion property and that surface tension of water, because we all observe with our eyes already how insects could walk on water. It's because of this large numbers of the water molecules and the hydrogen bonding gives it a strong structure and gives it that high surface tension, right? So if you look at the third point there, hydrogen bonds contribute to the many properties water molecules have that make them so important. Example, solvent, right? Because of its polar nature, it could dissolve most substances in the world, most solutes, right? High specific capacity, high, and we already know that it could take in great amount of heat before it changes state. High latent heat vaporization, we also know that too. So those two um, terms there is from CSEC physics, right? And water is less dense that when it's a solid, there is an anomaly, there's a unique property of water because we know when most things are in solid, they are more dense than when they are in a liquid or gas. But there is a reverse for water, right? And water has a high surface tension and cohesion. And we heard that cohesion word already in CSEC biology with transpiration and how water moves up the xylem, right? And it acts as a reagent, right? So the preceding slides here will just explain these to an extent, right? So we have hydrogen bonding there. So what is needed for hydrogen bonding, right? What is needed is a electronegative atom, which is oxygen, and you need to have your lone pairs of electrons, and you have your hydrogen. So you have your hydrogen bonding there between an electronegative and the hydrogen, right? Important structure, right? And we have what will be tested in your cape is to explain the properties of water with its structure, right? So water has many essential roles in living organisms due to its properties, the polarity of water molecules, right, and the presence of a number of hydrogen bonds. So these two things, the polarity, and we know how the polarity came up with oxygen being electronegative and pulling electrons, and you have that uneven distribution of the charge. And the hydrogen bond in there, well, it was a good candidate for that. So it could have that hydrogen bonding amongst itself within its structure, right? So solvent, the first property is the universal solvent, right? Because of that polar nature, right? And we already know from CSEC chemistry that ionic substances, CNA sodium chloride, is also considered polar because you have the positive, you have the electrostatic attraction of electrostatic positive and negative charges, right? So you'll find that salt, which is ionic, dissolves well in water, right? Because of that principle that we were taught in CSEC chemistry, like dissolves like, right? So water is polar. It has partial negative and partial positive ends. And you have ionic compound where you have the opposite charges there. So like dissolved like, right? So you see in your points here under solvent, this allows chemical reactions to occur within cells, right? As the dissolved solutes are more chemically reactive when they are free to move about, and the metabolites can be transported efficiently. So water being a good solvent, it helps with these reactions and transportation of substances within an organism, right? This is just showing you your polar molecules. There is a sodium there, plus ion and a chloride ion being surrounded by water. And it could dissolve because like dissolves like, right? But you have your water, um, which is polar. It can dissolve an organic substance, right? Like that what you see around in the blue there. So if ever in tape they ask you to explain, well, you know the structure of water already, right? That bend shape, and you know how the hydrogens are bonded covalently, and you know how the charges are distributed to give this polar nature. You know the hydrogen bonding diagram. After that, they might ask you, explain two properties of water. And if you talk about the solvent one, or you could talk about the high specific heat capacity. And it's basically the principle why it has a high specific heat capacity, great amount of heat before it changes a state, is because of those hydrogen bonding. And we talk about when the hydrogen bonds are small, well, it's weak. But if it is, you have plenty hydrogen bonds in a structure, well, then it will take great energy to break them, right? And you see any advantages, there's one more. You see any advantages for high specific heat capacity, provides a suitable habitat, right? It maintains a constant temperature in the body, right? So that the enzymes can work effectively. And also in those habitats too, you wouldn't want the water changing ever so often by great temperatures, then the homeostasis of the organism, they can't control that and they might die. So because of that high specific capacity and which is due to the many hydrogen bonds in water, it leads to these advantages for living organisms, right? And the latent heat of vaporization again is based on the hydrogen bond. So explaining the properties of water will be based on the hydrogen bonding and polarity. Well, these two here is based on the hydrogen bonds, right? You look at the last point for latent heat of vaporization. This provides a cooling effect for living organisms, for example, transpiration for leaves, or evaporation of water in sweat on the skin, right? So these two properties are based on the hydrogen bonding that water is involved in, right? So this table here is basically showing you the property of a universal solvent of water is based on the polarity of water. The high specific capacity is based on the presence of many hydrogen bonds. And the high latent heat of vaporization is based on the presence of the many hydrogen bonds. Right? And that coolant there is like in your radiator of your vehicle. You could ask your father about that, okay? How that coolant keeps the engine running. Right? So, and then, so that was it for water. Remember, my notes and my explanations will be basically concise with the objective of the syllabus. Because remember, CAPE is a lot of content crammed in 3 terms, right? So when I teach these objectives, I tend to go with the objective of the syllabus and the past paper, what normally comes for that objective, and give the students that explanation and note. So all for water, structure, why it has the polar nature and hydrogen bonding, and then to explain your properties, right? And you saw your table, your summary table there. Solvent is with the polarity, and the heat capacity and latent heat of vaporization is the number of hydrogen bonds. Good. Now your biological molecules, which are many that you have to study, right? Your carbohydrates, which is your broken down into your monosaccharides, your disaccharides, your polysaccharides, right? Then you will have examples of your disaccharide with your sucrose. We had to explain that to the structure and function. Uh, your starch, your glycogen, triglycerides, phospholipids, proteins, amino acids, right? So you learned a little bit about this in CSEC biology, right? The composition of each one of these, right? So what is just new is the full structure, because in CSEC chemistry, we use some simple diagrams for representation of glucose and so on. But now you're looking at the full structure of these compounds, right? So firstly, we want to look at, remind ourselves from CSEC chemistry, what is a polymer? Remember, that is a macromolecule, right? Long chain compounds form from repeating units called monomers. And if you look at your lipids there, which is fats, the monomers will be your fatty acids and glycerol, right? Those will come together and form your triglyceride, right? And your nucleic acid there, we see it again, That is with your pentose sugar, nitrogenous base. Once those come together, it will form your nucleic acid. Right? Same thing for carbohydrates. It will be various monosaccharides or disaccharides coming together as the monomers and forming your various carbohydrates. Right? And for proteins, will be amino acids coming together to form your protein. So this is just showing you again the amino acids, the monomers coming together to form your polypeptides. Many polypeptides will form your proteins. Monosaccharide coming together to form your disaccharides. Many disaccharides form your polysaccharides and so on. Right? So it is we coming into your molecules, your biomolecules, and we're looking at polymers because most of, if not all of your biomolecules are polymers. So we want to understand the formation of polymers and then the breakage of polymers, because the breakage of polymers like your carbohydrates, your proteins, your fats or lipids will be in digestion where it goes back into the monomer, your simple molecules before absorbing into the blood. Right? So first one we want to look at here is the carbohydrates. So from CSEC biology, you already have the first line there, made up of carbon and hydrogen and oxygen, right? So, and you remember from CSEC chemistry, you know that carbon is surrounded by four. It has four. It could facilitate four bonds, covalent bonds, right? And it could accommodate oxygen, nitrogen, sulfur. It could form straight chain, branch chains, or rings, right? We that first 2 points there is from CSEC chemistry, right? And also what is monomers and polymers and macromolecules, CSEC chemistry again, right? So again, CSEC chemistry there again, you're seeing made up of your C, your carbon, your hydrogen, oxygen, your ratio, and then you have your types of carbohydrates, your monosaccharide, your disaccharide, and your polysaccharide. All of that you already know, right? The new stuff, I'll tell you what is new, right? So these are the types of carbohydrates. Your monosaccharide is a single sugar monomer, right? All reducing sugars. Your disaccharide is formed from two monosaccharides joined by a glycosidic bond. We'll look at that today, right? And yeah, and the type of reaction involving that condensation. And a polysaccharide is the polymer formed by many monosaccharides joined by the glycosidic bond, right, in a condensation reaction. And you know condensation reaction from CSEC chemistry as we omit in water, right? And you see examples of your monosaccharide, your glyceraldehyde, right, 3 carbons, your ribose, yeah, 5 carbons, glucose 6 carbons, right? And you see in this alpha and beta in the disaccharide, we come in to talk about alpha and beta glucose, right? And the ring structure. That is new, right? So everything so far with water, you already knew most of that from CSEC. And what you're seeing here now is some structures for the examples of your mono, di, and polysaccharides, where you have to understand the structure, not the full structure. No simple structure again, right? And then you see the function there for each one of them. So this here is important. This here, if you look at a K-pass paper, you would see glucose is tested in structures and asks in certain things, right? So the most well-known carbohydrate monomer is glucose. Glucose has the molecular structure. We know that from C6H12O6. And you have your different types of your monosaccharide, your triosis, 3 carbons, pentosis, your 5 carbons, and your hexosis, 6 carbons. That is where you are accustomed with in C-set glucose, right? Glucose exists in two structurally different forms, alpha glucose and beta glucose, and therefore known as an isomer. So these two alpha and beta are known as isomers in C-set chemistry is same one. It will be, I'll show you saying it over in your head there, right? It'll just be same molecular formula but different structural formula, right? So it is basically, when we look at these, you'll see that they differ structurally, right? So the structural variety results in the different functions. And look at that. When we look at the structure of the alpha and the beta, just by the position of a certain atom, it will have a different function, right, in living organisms. So this is showing you the straight chain and ring structural formula of alpha and beta, right? If you look at it here, you would see 1, 2, 3, 4, 5, 6, right? And your age 12 and your O6, right? So this is your glucose straight chain, and you're seeing your hydroxyl group, carbonyl group, right? When in aqueous solution, glucose forms a ring structure. So once you put it in the aqueous solution, forms a ring structure. And most of my C6 students would have saw me drew this when we was looking at polymers in C6 chemistry, right? And then we went to the simple diagram fit. So you think about this here now, where you hold it up like so, and where you see not OH is saying below the ring and above the ring. So that yellow one would be like so, and this purple one will be like so. So you think about the ring like this, and you would have that yellow OH up in the air like that. So you'll have all the others around the whole hexagon there, flat, right? But you'll have this here yellow one upwards, and this pink or purple one there is on the underneath, right? And that there is the alpha here. And look at the number of it. Is the OH on number 1 and UH on number 3. Look at the OH there again in beta is on number 1 and number 3 carbon. So again, for the beta, you would be seeing that number 1 now on top. And the number 3 OH will be also on top. So you have the alpha with it alternate top and bottom. So 1, 3, 1, 3 is the alpha. And with your beta, you have them both on top. 1, 3, 1, 3. Right? So that is basically, you'll have to differentiate or distinguish that in your K paper or draw it. Right? And this one here is talking about starch is a polysaccharide, glycogen is a polysaccharide, cellulose. We have to study all of these molecules and its functions and structure later on, right? So what monomers they made up of? Well, the starch is only made up of your alpha glucose, your glycogen alpha, and your cellulose your beta, right? And you remember your alpha is your alternate, 1, 3, and your beta is 1, 3. Got them on top, right? This one here is just to explain covalent bonding a little bit again, because when you have the monomers coming together, the fundamental bonding there when they omit in water and join in is covalent bonding, right? This was just to remind ourselves the fundamental bonding um, in your formation of your polymers. And we all know this from CSEC chemistry, where you'll have your monomer coming together, two more monomers coming together, emitting water, and forming a polymer and a molecule of water, right? Condensation reaction. And when, like your carbohydrates, which is a polymer, your protein, which is a polymer, your fats, which is a polymer, goes into your digestive system, it is broken down by enzymes and water. Um, and that process is hydrolysis. And we call it in CSEC digestion, right? But if we go down to the technicalities, this is hydrolysis. You're adding back water now to get back your monomers, right? This is just showing you some covalent bonds in the organic molecules. So your carbohydrates is a glycosidic bond. We're going to look at that just now. Proteins is a peptide. Lipids is the ester. You know that proteins and the lipids one you know from CSEC chemistry. And your phosphodiester and your glycosidic is two new bonds, right? We all know this from CSEC chemistry with your reducing and non-reducing sugars and your oil rig, right? So reducing sugars donate electrons, non-reducing cannot donate electrons, right? And we have your formation of your glycosidic bond, right? To make monosaccharides more suitable, because remember, your monosaccharides will be more or less soluble. And if they are being transported, they will tend to be used up. So in order to make them able to be transported and stored and less affected by osmosis, they are converted into disaccharides and polysaccharides. And that bond that is formed among your monosaccharides into when they go into disaccharides and polysaccharides is the glycosidic bond, right? So looking at it there, the second point, um, form a strong covalent bond for here. That is where the two hydroxyl groups um, on different saccharides form a strong covalent bond called a glycosidic bond, right? And we see it here much more better. We see in, if you look at this here, you have your glucose and you will have condensation, your water coming out, and you'll be forming this 1,4 COC glycosidic bond, right? So once this H come out here and this OH, and you form, so these monosaccharides coming together to form a disaccharide. So you add 2 alpha, right? 2 alpha glucose coming together to form your disaccharide, which is maltose. So there's a structure for maltose, right? And your 1 water molecule omitted. Each glycosidic bond is catalyzed by an enzyme specific to which OH group you're interacting. As there are many different monosaccharides, this results in different types of glycosidic bonds. Your maltose is defined as your alpha 1,4 glycosidic, is so in the previous slide, and sucrose is seen in the preceding slide, alpha 1,2 glycosidic. So we're looking at your alpha glucose and beta fructose. And once your water is omitted, you form your sucrose, right? So you see the formation there. So this is something for your maltose or your sucrose. I may ask you to draw the structures for that in your exam. Right? This is an next example. This is structure of amylopectin, a polysaccharide, a major component of starch, and you see the glycosidic there again, right? 1,6 glycosidic and a 1,4 glycosidic. So this table here is just showing you types of glycosidic bonds when you have various glucose or monosaccharides coming together to form your disaccharide. Maltose is the alpha 1,4, sucrose alpha 1,2, cellulose beta 1,4, amylose alpha 1,4, amylopectin alpha 1,4 and alpha 1,6. Right? And the purple there is your disaccharide, your blue there will be a polysaccharide. Breaking a glycosidic bond, you add water, is hydrolysis, right? Because it comes just like with your polymers being broken down with water in the digestive system. So the same thing, right? So once water is added, you get back your monomers. Look at this one here. You have your glycosidic bond, maltose, and you add water, and it form back here alpha glucose and your alpha glucose. Again, the same thing there with your sucrose, water, and you get back your alpha glucose and your beta fructose. Right? So I hope everybody you benefited from this video. You got some, if it's one line or a diagram to augment your notes, that is the objective of the video, right? To assist you to ensure that you're covering your material, you're understanding it, and you're moving forward. Because remember, CAPE is a lot of content and in a short space of time, right? So the preceding videos, I would do the same thing and explain your sucrose, starch, glycogen, triglycerides, your phospholipids, your protein, with their structure and function within the organism, and then you test for certain biomolecules, and then I will recap with some probably summary notes, right, in preceding videos, right? It all depends on your comments, if it is required, right? So I hope it was helpful. God bless. Take care.